So let's discuss the breakdown of glucose in the presence of oxygen. So now we're actually going to move into the mitochondria. So up to this point, we've been discussing what happens outside the mitochondria. So we've been looking at glycolysis and how we convert glucose into pyruvate. And if we don't have oxygen, how pyruvate can undergo another process called fermentation. And so if oxygen is present, now we can move into the mitochondria. So here is our mitochondria right here. So we can move into mitochondria and start to further make energy. So inside the mitochondria itself is where we have three different phases that can take place. So again, if we're moving into mitochondria, we actually have the presence of oxygen. So while glycolysis occurs outside the mitochondria in the cytoplasm, inside mitochondria, the three phases are the preparatory reaction, the citric acid cycle, and the electron transport chain. So the next state of this glucose breakdown is the preparatory reaction. So we're now moving in to the mitochondria because there is the presence of oxygen. And so recall that previously we had converted glucose, which is a 6-carbon sugar, into pyruvate, which is a 3-carbon sugar. So now we have 3 carbons here. So what's going to happen in this state is that we're going to take the 3-carbon and convert it to a 2-carbon. So we have 2 over here. So in order to do that, this pyruvate is going to be oxidized, and in the process of oxidizing, remember that oxidation means that we're going to release electrons and we're going to also release hydrogen atoms, right? So we're going to release this electron hydrogen atom, and those will be added onto NAD plus to make NADH. The carbon that was released will become carbon dioxide. So that's the carbon dioxide that we actually breathe out. So now we have a 2-carbon atom here, and we're going to attach that 2-carbon atom to coenzyme A, resulting in acetyl-CoA. So what is the end result of the preparatory reaction? The end result of the preparatory reaction is to take the 3-carbon molecule pyruvate, convert it to a 2-carbon, add it to acetyl-CoA. This happens twice because recall that we went from 6-carbon to 3-carbon. So it is going to produce 2 carbon dioxide molecule, 2 NADH molecule, and 2 acetyl-CoA molecule. All right, so now that we have our acetyl-CoA, we can go ahead and move on to our next step, which is going to be the citric acid cycle. So now that we have our 2 acetyl-CoA molecules, we're now ready to enter the citric acid cycle. So this is still occurring in the mitochondria, but now we're in the matrix of the mitochondria, and the acetyl-CoA is going to be transferred to a 4-carbon molecule. So what we're going to produce is a 6-carbon molecule, also known as the citric acid. The purpose of the whole citric acid cycle is basically to transfer electrons and hydrogen atom to NAD plus and FAD. NAD plus and FAD are both electron carrier. So our goal is to actually make these molecules that carry electron that we're going to use later on in the glucose breakdown when we get to electron transport chain. The citric acid cycle takes many different steps, and each step is an oxidation reaction where we're going to oxidize the starting product into another product, and in the process release electrons and hydrogen atom that are going to be added onto our electron carriers, as well as releasing a byproduct of carbon dioxide. So let's start off on the top of the cycle. So recall we have acetyl-CoA, which is going to be a 2-carbon molecule added to a CoA enzyme that's going to be added to a 4-carbon molecule. That 4-carbon molecule, as we mentioned, plus the 2-carbon molecule will make citric acid, which is a 6-carbon. So now we have this 6-carbon molecule. We undergo our first oxidation reaction that's going to release electron, hydrogen atom, those will be added onto our NAD+, and we release CO2. So 6 minus CO2 is a 5 carbon. So now we have a 5 carbon molecule. We repeat that again, where we do an oxidation reaction, and we end up with a 4 carbon molecule. Now we're going to undergo another series of oxidation reactions. So we're still doing oxidation reaction, but rather than converting and the molecule and losing carbon dioxide in the oxidation reaction, we're going to basically convert these different molecules and rearrange them. And in the process of rearranging, we're still doing oxidation reaction, but as we're rearranging, we're releasing electrons that can then be transferred to these other electron carriers. So we form FADH2 as well as NADH. We also are able to do a substrate level ATP synthesis. So that means that we're actually going to produce an ATP as well. So in this process of the citric acid cycle, the outcome of this is not only producing 2 molecules of ATP, but we also produce all of these molecules, electron carrier, that now contains an electron that we can then carry on to our next step. Right, so let's revisit the output so far of what we have. So as I mentioned, the citric acid cycle, we start off with acetyl-CoA, and we're going to undergo the cycle 2 times, right? Because we have 2 molecules of acetyl-CoA. So our input is 2 molecules of acetyl-CoA. We also are inputting all these different electron carriers as well as ADP, which we later will convert through the series of oxidation reaction into NADH, FADH2, and ATP. So the output of this is producing some carbon dioxide byproduct from when we broke down the molecules into smaller molecules from oxidation reaction, as well as the transfer of electrons and hydrogen to our electron carrier. So now that we have FADH2 and NADH, and the substrate level ATP phosphorylation, which results in 2 ATP. All right, I'll put this video up onto our YouTube site so that you guys can watch this video if you're interested in it. And let's kind of recap before we move on to our next step and see if we can answer these questions. So the first one is, how many carbons are left at each stage of the citric acid cycle, and how many carbons are in glucose to pyruvate to acetyl-CoA to carbon dioxide? How many net ATPs are made at each stage between the glycolysis, the preparatory reaction, and the citric acid cycle? And the last question is, how many coenzymes, right, so these electron carriers I've been talking about, of NADH and FADH2 are made at each stage? So again, up to this point, we've talked about glycolysis, the preparatory reaction, and the citric acid cycle. So pause the video and see if you can answer these questions on your own, and then we'll go through them. Okay, so let's talk about the first question, which is asking how many carbons are left at each stage. So we start off with a 6 carbon molecule, right? That's our glucose. And the first stage is glycolysis. So if you recall, glycolysis is going to convert that 6 carbon into 2 3 carbon. So that is the purpose of it. So the end result of that glucose converting to pyruvate is 2 3 carbon molecules, which means that at the end of this reaction, we still have 6 carbon. The next stage is the preparatory reaction. So the goal of this preparatory reaction is to convert the pyruvate into acetyl-CoA. That's our end goal, right? So we're going to take the 3 carbon molecule and convert it into a 2 carbon molecule that then we add on to coenzyme A, forming acetyl-CoA, and we release in that process a CO2 molecule. We do this twice because we have 2 molecules of pyruvate. So remaining is 4 carbon, right? 2 carbon plus acetyl-CoA twice would be 4 carbon. So now from the preparatory reaction, we move on to the citric acid cycle. So we're going to take our acetyl-CoA, which at this point has 2 carbons, and we're going to enter the citric acid cycle twice. In that cycle, we're going to undergo a series of oxidation reactions. Those oxidation reactions is going to end up resulting in CO2 being released as well as NADH and FADH2 being produced, which we'll talk at a later point, as well as ATP, which we'll also talk in the next question. So the number of carbon remaining is 0 because we fully oxidized our acetyl-CoA when we underwent that all of those oxidation reactions, releasing carbon dioxide and transferring those electrons and hydrogen atom from our molecules to our coenzymes. Okay, so we went from glycolysis, results remaining is full 6 carbon because as 2 pyruvate molecules. Preparatory reaction, we have 2 acetyl-CoA molecules, and so our remaining number of carbons right there is 4 carbons. By the time we get to the citric acid cycle, we've released 4 carbon dioxide, so we lose all 4 carbons that we started with, and so the carbon remaining is 0. Okay, so let's move on to question number 2, which asks us how many net ATP are made at each stage. Okay, so let's start with glycolysis. So glycolysis, we produce 2 substrate level ATP, right? But remember that we took some ATP in order to convert that glucose into pyruvate. It also took some ATP, but our net result is still 2 ATP. The preparatory reaction, when we were converting our pyruvate to acetyl-CoA, there was no ATP made, so 0. And in our last step, which is the citric acid cycle, each time we went through the cycle, if you recall, we made 1 ATP molecule, which means for every glucose, we make 2 ATP. So far, up to this stage, we've made 4 ATP possible, 2 from glycolysis and 2 from the citric acid cycle. Move on to question 3, which asks about the coenzyme or electron carriers. So how many coenzymes, either NADH or FADH2, are made at each stage? So our first phase was glycolysis, so converting that glucose into the pyruvate. If you recall, we not only were able to produce substrate level ATP, but we were able to make 2 NADH per every glucose molecule. So now we enter the preparatory reaction. We have 2 pyruvate. As those pyruvate are being converted to acetyl-CoA, we produce 1 NADH. So resulting in 2 NADH because we undergo this reaction 2 times. Lastly, let's look at citric acid cycle. So in the citric acid cycle, we are taking the acetyl-CoA, adding it to the 4 carbon citrate, and then undergoing this cycle, which is a series of oxidation reaction. As we undergo this series of oxidation reaction, it's being coupled with the reduction of NAD plus and FAD in order to make NADH and FADH2. So we actually, in total, produce 6 NADH per glucose because we undergo the cycle 2 times, right? So for each acetyl-CoA, when we undergo it 1 time, we produce 3 NADH. Do it twice, therefore we have 6 NADH. We produce 1 FADH2 per acetyl-CoA. We undergo this twice, so the result is 2 FADH2. So total number of coenzyme or electron carrier that we produce through these 3 phases is 10 NADH per glucose molecule and 2 FADH2 per glucose molecule. All right, so on our next talk, we'll be actually starting to look at the electron transport chain. I'll see you there.